Hello, this is video number 2 for module 6. In this video, we're going to look at a diagnostic cross that you can do to determine if two genes are linked or unlinked. And this is in the situation for autosomal genes. Later on, we'll talk about the special circumstance for X-linked genes. So just taking a look at the outline, we're talking about number 3, linked genes tend to segregate together in meiosis. And like I said, special case of autosomal genes. And we're going to look at the situation for what's called complete linkage versus incomplete linkage. And we're going to compare it to what we talked about on the previous slide, which was the situation for unlinked genes. All right, so this slide is showing the diagnostic cross for two genes that are unlinked. And this is a dihybrid test cross. We're starting with the parental generation. An individual that's homozygous, pure breeding for two different genes. Are they linked or unlinked? We don't know. So to determine that, we're going to set up a test cross. Here's the tester individual, homozygous recessive for both genes. The F1 is going to be heterozygous for both A and B. And then here is the diagnostic cross. Cross the F1, double heterozygote to a tester. It's a double heterozygote test cross or a dihybrid test cross. Either of those would be correct. And this would be the result that we talked about in the last video. From this doubly heterozygous individual with genes on 2 chromosomes, right? We're assuming they're unlinked. All 4 gamete genotypes are present in equal frequency, and that would produce all 4 offspring genotypes in equal frequency. 2 of these would be parental genotypes, the one showing both dominant traits, similar to this parent, and the one showing both recessive traits, showing similar to this parent. 2 of these are non-parental or recombinant types, being dominant for A and recessive for B, or recessive for A and dominant for B. Since each of the 4 classes are 25% each, the percent of recombinants is 50%, and the percent of non-recombinants is 50%. Okay? So that's the situation for genes that are unlinked. And this is the same table from the previous slide. The summary is, when 2 genes are unlinked, the diagnostic cross, the dihybrid test cross, shows 4 F2 phenotypes, 25% each. And the percent recombinants is equal to the percent of non-recombinants. All right, let's look at the situation for linked genes. And just a note, if we are assuming that genes are linked, a good way to draw them, their genotype is like this. So this would indicate one chromosome and the alleles on that chromosome. This would indicate the other chromosome and the alleles on that chromosome. In this case, one chromosome is carrying 2 dominant alleles, the other chromosome is carrying 2 recessive alleles. It doesn't necessarily have to be that way. It could be that this chromosome would be dominant for A and recessive for B, and this one would be recessive for A and dominant for B. Okay, but in this notation, we're using lines to indicate chromosomes. Okay, so here's an example of a cross in which the genes are showing complete linkage. And what complete linkage means is if you set up this cross, pure breeding for A and B, dominant traits for both, to pure breeding for little a and little b, recessive traits for both, you would get this F1 offspring. This F1 would get 1 chromosome with both dominant alleles here, and 1 chromosome with both recessive alleles. All right, if then we set up a dihybrid test cross to a tester individual, then what would we get out of that? So in this special case called complete linkage, there is no crossover between the A and the B gene. The genes are linked, but there's no crossover. If there's no crossover, the F1 double heterozygote will produce only two types of gametes, one with both dominant alleles or one with both recessive alleles. As a result, only two types of offspring would be produced, one showing both dominant traits and one showing both recessive traits. Both of these are parental or non-recombinant phenotypes. One parent, both dominant traits, the other parent, both recessive traits. These two types would be produced in 50% each, but they would both be non-recombinant types. So to summarize, for complete linkage, when two genes show complete linkage, the diagnostic cross, dihybrid test cross, only shows two F2 phenotypes in 50-50 proportions. And all F2 progeny are non-recombinants. Okay, what's the situation? Possibility number 3, shown on this slide. Let's say that there are two genes that are linked, A and B, but they are far enough apart in the chromosome that crossover can occur for them. In the previous example, for complete linkage, the genes were extremely close, and there's never crossover because they're so close together. But in the case of incomplete linkage, the genes are far enough apart and crossover can occur between them. So if you cross a pure breeding individual containing both dominant traits to a pure breeding individual with both recessive traits, you'll get an F1, doubly heterozygous, and this F1 in some meioses will have crossover between the A and the B gene. That will produce some gametes that have little a with big B or big A with little b. Those will be crossover or recombinant gametes. In addition, there will be some gametes that have the parental combination, big A with big B, and little a with little b, right? Parental generation had both of those, but neither of these two. Okay, what would be the result of that cross if we look at it in Punnett square? Well, from the doubly heterozygous individual, some of the gametes would be this parental type, both dominant alleles, or this, both recessive alleles. Some of them would be recombinants or non-parental, big A with little b, little a with big B. Since we're doing a test cross, that would be reflected in the offspring as well. Now, for genes showing incomplete linkage, there is some crossover between the linked genes. What we would see in terms of percentage is the parental types would be more common, and these two types would be equal in frequency. So here's an example. They could be 40% each, but this can vary. This number can vary. The recombinants or non-parentals would be less common, and these two types of recombinants would be equal in frequency. So if these two are 40%, these two would be 10%, half of the remaining 20%. Okay? So in summary, when two genes show incomplete linkage, the dihybrid test cross shows 4 F2 phenotypes in unequal proportions. The parental phenotypes are equal to each other and present in more than 25% each, more than 50% together. The two non-parental or recombinant phenotypes are also equal to each other, but together some less than 50%. All right, I think I'll end this video here, and in the next video, we'll talk about the consequences of crossing over and calculating a frequency of crossover.